Okay, so welcome to this next video in the playlist on G protein coupled receptors. In this video, what we're going to talk about is heterotrimeric G proteins. So we're going to discuss firstly the structure of heterotrimeric G proteins. We're then going to discuss the lipid modifications that occur to heterotrimeric G proteins, which allow them to anchor themselves onto the lipid bilayer, basically. Okay, and then what we'll discuss is the G protein cycle, so how G protein coupled receptors then activate heterotrimeric G proteins, and how heterotrimeric G proteins then inactivate because the alpha subunit has a GTPase within it. Okay, so we'll start off then with the basic discussion of the structure of heterotrimeric G proteins. So basically, if this is the plasma membrane, let's say here, okay, so these two lines represent the outer and inner leaflet of the phospholipid bilayer, then we have this complex known as a heterotrimeric G protein that binds to the inner leaflet of the phospholipid bilayer, and the reason it can do this is that it has lipid molecules coming off the protein subunits, okay, and these lipid molecules implant into the lipid bilayer and therefore anchor the heterotrimeric G protein at the inner leaflet of the phospholipid bilayer. Okay, so heterotrimeric G protein. Hetero means different. Trimeric means three subunits, okay? So these are structures which are made up of three separate proteins that are all different. So let me now show you these. So one of the key subunits that you have is the alpha subunit. In fact, potentially the most important subunit. Arguably, certainly the most important. Okay, so I'll draw it like so. Okay, and I'll label it the alpha subunit, and I might even colour it in, it's so important. Okay, and basically the alpha subunit will have a lipid molecule attached to it. Now some alpha subunits will actually have two lipid molecules attached to it. We will discuss palmitoylation and myristoylation later in more detail, but for now I'll just draw one lipid molecule. More generally, most of them just have one, but some of them, as we'll see later, actually have both a myristoyl group and a palmitoyl group coming off. So they have some lipid molecule that implants into the inner leaflet of the phospholipid bilayer and holds the alpha subunit at the inner leaflet of the phospholipid bilayer. So this is how it attaches to the inner leaflet of the phospholipid bilayer. Okay, now the alpha subunit, when it's in the inactive stage, has guanosine diphosphate linked to it. Okay, so it's bound to GDP, guanosine diphosphate. And this is a general feature that goes for all G proteins, not just heterotrimeric G proteins, but also monomeric G proteins, which we're not really going to discuss in any great detail here. But the general principle is that there are two states, basically. It's like a switch. It can either be on or it can be off. And in the off state, it has guanosine diphosphate bound to it, and in the on state, it will have guanosine triphosphate, GTP, bound to it. Okay, so currently it's in the off state. Now in the off state, what happens is the alpha subunit binds to two other subunits known as the beta and the gamma subunit. So let's draw these here. So basically, beta and gamma always remain bound to one another, and when the alpha is in the off state, they'll also be bound to alpha. Okay, so I'll show this here. So you have here is the beta subunit, and then the beta subunit is bound to the gamma subunit, and they always remain bound together. Okay, but when the alpha subunit is in the off state, this complex, which is called often the beta-gamma complex, will associate with the alpha subunit, so they'll bind together. Now the beta subunit always has to remain in association with the gamma subunit because it's the gamma subunit that has the lipid modification attached to it, and this is a different type of lipid modification to the lipid modification that you have for the alpha subunit. The alpha subunit has either a myristoyl group or a palmitoyl group, or both potentially attached to it, and we'll discuss myristoyl and palmitoyl groups later in much more detail, so don't worry if you don't know what they are. This is really for people who already know about lipid modifications of proteins. And the gamma subunit has something known as a prenyl group attached to it, and we will discuss this in a lot of detail later. Okay, so let me colour in the different subunits. So the importance of the beta subunit binding to the gamma subunit and always remaining bound to the gamma subunit is that without the gamma subunit, it has no means to attach to the inner leaflet of the phospholipid bilayer. It's the gamma subunit that has the lipid group which anchors into the lipid bilayer and holds the gamma subunit at the inner leaflet of the phospholipid bilayer. And the beta subunit only stays there because it's bound to the gamma subunit which is staying there. Okay, so in the off state, what will happen is that the alpha GDP molecule here will associate and bind with the beta gamma complex here. Okay, right, now before we go any further, I want to discuss with you all the different types of alpha subunits, all the different types of beta subunits, and all the different types of gamma subunits. And this is where it gets a little bit heavy. Okay, so basically, in the human genome, as of yet, and this is evolving all the time basically, but as of yet, and it's remained constant for quite a few years now, I think almost a decade now, so hopefully this is actually right what I'm presenting to you. But at present, what we believe is that there are 16 genes for the alpha subunit, okay, in the human genome. Okay, so there are 16 different genes which all code for alpha subunits, but it gets more complicated than that. So this is the alpha subunit that we're talking about at the moment. Because there aren't 16 different alpha subunits, there are actually 21 different subunits, okay? So there are 21 different types of alpha subunit. So this doesn't seem to add up. There's 16 genes, but 21 different subunits. How is that happening? Well, basically, it's because there are different splice variants, basically. So let me just give you the little principle of splicing. Okay, so let's say we have our DNA here, and to make the picture look simple, I'm not going to show it twisted around. So here are the two strands of DNA, okay? Now, let's say we have here our gene for one of the alpha subunits, okay? So I'll pick the one that generally people have all heard of, which is the G alpha S subunit. If you haven't heard of it, don't worry, we'll see it in a moment, okay? So let's say this is the gene for the G alpha S, and of course you'll have two genes. You'll have one from your mother, one from your father because of the homologous chromosome system. But let's just say this is one of them, okay? And basically what will happen is when you want to actually make the protein that corresponds to this gene, what will happen is RNA polymerase II will come in, open the DNA up, it will make a piece of mRNA, so let's show that here, which is complementary to one of the strands, okay? And the strand which you make the mRNA complementary to is called the coding strand. So let's say this one that I've just highlighted in turquoise here, this is the coding strand of DNA. Okay, and basically what we will do is we'll make the mRNA complementary to this coding strand. So then the piece of mRNA will come out, and it will be an exact replica of the non-coding strand, basically. So it will have the same combination of organic bases as the non-coding strand, which, remember, was complementary to the coding strand. So the other strand, which isn't used for transcription, is called the non-coding strand. And of course, the non-coding strand is complementary to the coding strand, and we've also just produced this mRNA which is complementary to the coding strand, so basically they will be the same sequence of organic bases, with the difference, of course, that you don't have thymine organic bases in mRNA, instead you replace them with uracil. Okay, but that's the only difference that you'll have in the sequence of organic bases. Okay, so here is our piece of mRNA. Now, the question is, do you use this piece of mRNA to translate? Does it go straight through the ribosome and actually create a protein? No. This is called pre-mRNA. It's known as the primary transcript, basically. It's what you produce directly from transcription, okay, but you do not use this piece of mRNA yet for actual translation. Before it goes for translation, you have to cut loads of bits out. Okay, so you have to make modifications to this, and the modifications are splicing. So let me explain. Basically, there are portions of this piece of mRNA that you actually do want to translate. Okay, so let's say this little portion that I've shown here, this portion, is a portion that actually does need to be translated. It's part of the actual mRNA that needs to be translated to produce a sequence of amino acids that you want in the G alpha S protein. Okay, that's called an exon then. Okay, now there are other portions that are not necessary to actually include when you're actually translating. Okay, and these other portions are known as introns. So let's say here is another exon here, and the portion in between we don't need basically. If we translated this, we'd get some junk sequence of amino acids that's not functional basically. So you don't want to translate it. It's called an intron. Okay, and people often refer to them as the junk DNA basically. So, in this pre-mRNA, you'll have loads of different exons. So here's one exon, here's another exon, here's another exon. And let's say that's enough, basically. All the other bits in between are introns. Now, basically, the concept of splice variants is the concept that maybe there are some exons that you don't actually have to include. For instance, let's say that exon 1 and exon 2 here, so let's label these exons up. Exon 1, exon 2, exon 3. Okay, so let's say exon 1 and exon 2 are absolutely essential to make a functional G alpha S, but then exon 3 might be less essential. So you might be able to have it or not have it. Now that's quite an extreme example, but you do see that in nature, that you do see exons that you can include and you can choose not to include them. More commonly though, you see examples where you can cut bits off the exons, and that doesn't matter basically. So you can splice the exons in different ways basically, because if you imagine what's actually happening here, all that's happening is enzymes are coming and cutting the DNA at certain places. Now, are those places set? Well, reasonably, but potentially it can cut it at a slightly different place, producing you a slightly different mature piece of mRNA. So let me just draw the next piece of mRNA. The next piece will be where you stick all of the bits that you're actually going to use back together to create a piece of mature mRNA. So basically, from the splicing process where you're cutting out the introns and recombining the exons, you can actually get loads of different pieces of mature mRNA at the end of the process basically. And these different pieces of mature mRNA will then give rise to slightly different proteins, and these different proteins that come from the same gene are known as the different splice variants of that gene basically, or the different splice variants of that protein. Right, so basically, even though you've only got 16 genes, some of these genes can give rise to multiple splice variants, and G alpha S is an example where there are multiple splice variants basically, which is how you can get 21 different subunits even though you only got 16 different genes. Okay, right, so in the human genome you will have 16 different genes for alpha heterotrimeric G protein subunits. Okay, right. So, let's now discuss these 16 different genes. So they are classified into families, basically. And I also just want to stress, so that in case people are getting a little confused, I'm not talking about different alleles of the G alpha S subunit. Basically, if you count out your chromosomes, I'll put it here. Okay, so if we were to draw out your 46 chromosomes, like so. Okay, and it goes all the way on to the 46th one over here, right? Basically, these are different genes. These 16 different genes are at different points on the chromosomes. In fact, let me draw it a different way. Let me have the homologous chromosomes here. Okay, so let's say this is chromosome 1 to chromosome 23, and then you have the homologous chromosomes here. And then obviously 23 is a little bit more complicated because it can either be XX or XY. Let's say this is a girl, so it's XX. Okay, right. So this is chromosome 1, chromosome 2, all the way up to chromosome 23. We have the homologous chromosomes here. So basically, I am not just talking about different alleles of one gene. I am talking about separate genes at different points on different chromosomes. So basically, you will have 16 different genes that all code for alpha subunits. So here is one. Now, of course, you'll have the same thing on the homologous chromosome. So let's say this is the gene for, let's say, G alpha OLF, which is another example we'll see. Okay, and you've got two of those genes here. Then, if we go to another chromosome, let's say we've got another gene here for a different alpha subunit. Then we'll have the G alpha S gene somewhere. So we'll have one here, one here. So I am talking about different points at different chromosomes. Everyone will have all 16 genes for the different alpha subunits. I'm not talking about different alleles for just one gene. No. Okay. So I hope that's now clear. Let's now talk about the 16 different genes for alpha subunits. So they are classified up into 4 major families. Okay, and I'm not going to have space to put it up there, I don't think, so I'll have to come down here. Okay, so basically there is the G alpha S family of alpha S subunits, of G alpha subunits, okay? And this contains 2 genes. It contains the gene for G alpha S, okay, and of course you'll have 2 genes, so here they are for G alpha S, and then on another chromosome you'll then have genes for G alpha OLF, and of course you'll have 2 of these as well, one on each of the 2 homologous chromosomes. So, these are the 2 genes that are within this family of G alpha S, G alpha subunits, basically. Okay? Then the next family of G alpha subunits is the G alpha I/0 family of G alpha subunits. So this is just a classification for us to understand the different alpha subunits, and we categorize them together based on functional and also structural similarity. Okay, so the ones in each of the family will be structurally similar, and they'll also have similar functions, basically. So the G alpha I0 is the most complicated family. So this contains a lot of different genes. So firstly, there is the G alpha I1 gene. Then there is the G alpha I2 gene. Then there is the G alpha I3 gene. Okay, so each of these will code for slightly different alpha subunits. So you've got the G alpha I1 subunit, the G alpha I2 subunit, the G alpha I3 subunit. Then you have the G alpha T's. So you have G alpha T1, which is the alpha subunit that you have in rod cell transducin. Okay, so there is a heterotrimeric G protein that is extremely important in phototransduction in rod cells, and this is called rod transducin. And basically, the alpha subunit of rod transducin is going to be this G alpha T1 alpha subunit. Okay, you also have cone transducin alpha subunit, which is G alpha T2. So this is the alpha subunit within the heterotrimeric G protein that is cone transducin. Okay, so there are 2 main types of photoreceptors within the retina, rod cells and cone cells, and these are the 2 different alpha subunits that you have in the 2 different forms of transducin that you have in those 2 types of cells. Okay, you then have the G alpha 0 gene, okay? Now, this one is quite confusing, okay? There is only 1 gene for G alpha 0. Well, of course, you'll have the 2 on the 2 homologous chromosomes. But people, well, if you read the research literature, you'll often see people referring to G alpha 0 1 and G alpha 0 2. Now these are the different splice variants of this same gene. So from this one gene you get 2 different alpha subunits, but these are splice variants. Now this is the only gene where I'm actually going to show you the nomenclature for the alpha, for the different splice variants of this gene, basically. Other ones, such as G alpha S, have many splice variants, but the nomenclature is just hideous, so we're not going to go there. Okay, but this one shows up often, basically. Okay, then we've also got the G alpha Z gene, okay, and then finally in this family we've also got the G alpha gustatory, or just gust at the bottom, which is the one that's involved in transducing the sensation of taste. Okay, so those are our G alpha I naught family G alpha subunits. Okay, we'll continue this discussion in the next video with 2 more families of G alpha subunits of heterotrimeric G proteins.